you know, the next time you reach into the back of your fridge and you pull out that, like, totally forgotten orange.
Oh, the one with the fuzzy blue-green sweater on it?
Yeah, exactly. That one. I want you to just, pause for a second before you throw it in the trash. Or actually, the next time you're having a cold beer, take a second to really look at the frothy head on it.
Right, because that fuzz on your fruit and that foam in your glass, they actually share this secret and honestly pretty dramatic biological history.
They really do. Welcome to this deep dive, everyone. Today, we're pulling apart a massive stack of sources. We've got biological research, historical accounts of these really bitter 19th century scientific brawls, and even some modern epidemiological case studies.
Yeah, it's a lot of material, but it all points back to this history characterized by, bizarre, mistaken identities and organisms that essentially operate as microscopic shapeshifters.
Which is wild. So, the mission for today's deep dive is to completely shatter everything you think you know about yeasts and molds. We are exploring how these two fungal forms have shaped human health, how they spark foundational fights in biology, and why they absolutely aren't what they appear to be.
Definitely. And that's kind of a recurring theme in mycology, actually. Our visual biases have just led to centuries of fundamental misunderstandings about, well, what these organisms are even doing.
Okay, let's unpack this. And I want to start with the biggest misconception right out of the gate. Because when you hear yeast and mold, you probably assume they're like two totally different evolutionary branches.
They're right, like plants versus animals or something.
Exactly. But yeast club and mold club are completely fake evolutionary categories.
Yeah, they really are. What's fascinating here is that yeast and mold are strictly morphological descriptions.
Meaning they just describe the shape, right?
Exactly. They describe a physical growth strategy, not a taxonomic lineage. So identifying a fungus as a yeast or a mold gives you, honestly, zero information about its actual evolutionary family tree.
Wait, so you're saying two different molds could be completely unrelated?
Oh, absolutely. Two different molds can be completely distant genetic cousins, like significantly less related to each other than one of them is to a yeast.
Okay, so classifying all fungi as either a yeast or a mold is basically like grouping bats, birds, and bumblebees together just because they all happen to have wings and fly.
That is the perfect analogy. You're just categorizing them by a shared physical trait, like a lifestyle, rather than their actual genetics. And historically, relying on that visual shape caused massive taxonomic chaos.
I can imagine. I mean, we love to just group things by how they look.
Right. Like, go back to Carl Linnaeus in the 18th century. He was building the whole foundation of modern biological classification. And he looked at fungi, saw that they didn't move around and that they grew out of a substrate like soil. And he just said, okay, these are plants.
Which is a very human mistake, honestly. I mean, if it sits still and grows out of the dirt, it must be a plant.
Yeah, it makes total sense if you're operating without molecular genetics.
But it was entirely wrong.
Completely wrong. It actually took until 1969 when Robert Whittaker introduced his five-kingdom system for fungi to finally get recognized with their own independent kingdom.
1969, that is so recent.
Yeah.
And then modern molecular phylogenetics came along and delivered, like, the ultimate plot twist.
Yeah.
Because when you analyze the genetic data, fungi are classified as opistocons.
Opistocons, right.
Which means fungi share a much more recent common ancestor with animals, meaning with you and me, than they ever did with plants.
Yeah, the opistocon lineage is defined by a shared ancestor that had a single posterior flagellum, basically a little tail for swimming.
Okay.
And while fungi largely lost that flagellum over millions of years of evolution, animals actually kept it in specific cells, like sperm. But the molecular homology is totally undeniable.
So that mushroom popping up in your front yard is, genetically speaking, more closely related to you than it is to the oak tree it's growing next to.
Exactly, which means physical shape is just a terrible map for evolutionary relationships.
Right. So to actually understand yeasts and molds, we have to strip away the visual biases and look at them from first principles. If their family trees don't define what a yeast or a mold is, we have to look at what does define them.
And that really comes down to their physical strategy for survival. We can start with their shared baseline, the stuff they both do.
Okay, lay it on me.
So they are both eukaryotes, obviously, but unlike plants that use cellulose for structure, yeasts and molds build these really rigid cell walls using chitin and glucans.
Wait, isn't that what bugs use?
Yeah, exactly. It's the exact same complex nitrogen-containing polysaccharide that insects and crustaceans use for their exoskeletons. It gives them immense tensile strength, which they need to withstand massive internal turgor pressure.
Man, nature loves recycling a highly efficient structural polymer, doesn't it?
It really does. But the most critical shared trait between yeast and mold is how they acquire energy. They are absorptive heterotrophs.
Right. Meaning they rely on osmotrophy, they can't photosynthesize like plants, and they don't have an internal digestive tract like animals.
Right, so instead, they essentially turn the environment into their stomach. They excrete these hydrolytic enzymes directly outward into their surroundings.
To kind of digest their food on the outside?
Exactly. They use those enzymes to depolymerize complex macromolecules like breaking down big starches into simple sugars. And once the food is chemically dismantled outside their bodies, they use proton gradients to pump those nutrient monomers back across their cell membranes.
That is wild. So that's their shared biological engine. But the architectural strategy of how they deploy that engine is where yeast and mold completely diverge, right?
Yes,
So let's look at yeast first. The single-celled spender. It's unicellular, and it typically reproduces asexually through budding. Like a little daughter cell just pinches off as a perfect genetic clone.
Or, in the case of some yeasts like Schizosaccharomyces pombe, it happens through simple binary fission. But the really defining characteristic of yeast is sheer speed.
Right, they are fast. I read in the sources that Saccharomyces cerevisiae, which is just classic baker's yeast, it can form a visible colony from a single cell in just 24 hours.
Yeah, it's incredibly fast. And that rapid, simple division is actually why molecular biologists adopted yeast as the ultimate eukaryotic model organism. They were just so easy to cultivate in a lab.
And didn't that lead to a huge milestone in the 90s?
It did. In 1996, Saccharomyces cerevisiae became the very first eukaryotic organism to have its entire genome sequenced. Its simplicity made it like the perfect proving ground for genomic technologies long before we ever tried to tackle the human genome.
Okay, so we've got the single-celled sprinter. The alternative strategy is the mold, which we can call the filamentous forager. So mold grows via hyphae, right? Which are these microscopic branching tubular structures that tangle all together to form a mycelium.
Right, and the geometry here is brilliant because a spherical yeast cell has a highly constrained surface area to volume ratio, which limits how much it can interact with its environment.
Basically, a sphere only has so much outside surface compared to its insides.
Exactly. But by stretching out into these long, microscopic filaments, mold maximizes that ratio. A mycelium can penetrate super deep into a piece of rotting wood, for example, secreting enzymes and absorbing nutrients over this massive spatial area.
And some of those hyphal threads have internal cross walls, right, like septa, with tiny little pores that allow the cytoplasm and nutrients to just stream through.
Yeah, but what's even crazier is that some primitive molds are entirely coenocytic, meaning they skip the internal walls completely and just operate as one massive continuous tube filled with thousands of flowing nuclei.
Just one giant multinucleus cell tube. That's slightly terrifying. And for reproduction, they just launch massive payloads of airborne chlamydia or spores.
Yeah.
Which I guess is why that fuzzy mold on your bread seems to just magically teleport to your fruit bowl overnight.
Yes, spores get everywhere. And form dictates function perfectly here. I mean, consider Alexander Fleming's discovery of penicillin in 1928.
Oh, right. Penicillium rubens. That's a mold.
Exactly. It was specifically its filamentous hyphal growth strategy, like its ability to stretch out and excrete defensive compounds outward into the bacterial agar, that created that famous zone of inhibition that Fleming noticed.
Okay, but I'm going to push back on this narrative a little bit, on this idea of yeast being the simple one.
Oh.
Yeah, because if yeast is just this basic fast-dividing sphere, so rudimentary that we used it as practice for genomics back in the 90s, how can it possibly be a major medical threat? We always hear about mold spores being environmental hazards, but a single-celled yeast just seems too structurally basic to be a serious villain.
Well, that is a dangerous assumption to make, because in biology, simplicity often equals adaptability.
How so?
Look at Candioris. This is a yeast that was first identified in Japan around 2009, and today it's currently one of the most alarming nosocomial-meaning hospital-acquired pathogens globally.
Wait, a yeast is a hospital superbug?
Yes, and because it's a single independent cell, it doesn't need to weave this delicate mycelium to survive. It can just colonize human skin and persist on cold, sterile hospital surfaces for weeks.
Wow, so think about the last time you leaned on like a plastic armrest in a hospital waiting room.
And they
This yeast could just be sitting there waiting.
Exactly. It operates with the rapid transmission dynamics of a dangerous bacterium. And furthermore, it easily forms really dense biofilms, and it frequently exhibits intrinsic resistance to multiple classes of antifungal medications.
Like it just ignores the drugs.
Pretty much, echinocandins, ozoles, you name it. Its single-celled form is precisely what makes it so elusive and difficult to eradicate in a clinical setting.
Man, that is a terrifying level of efficiency.
Mm-hmm.
So yeast and mold represent two distinct biological strategies for dominating an environment. But here's where it gets really interesting.
The shapeshifters.
Yes. What happens when an organism figures out how to utilize both strategies? You get dimorphism.
Right.
I like to think of them as microscopic werewolves.
I love that.
Right, because there is a whole class of fungal species that possess a genetic switch, allowing them to shift seamlessly between the yeast form and the mold form, based entirely on environmental triggers.
Yeah, Candida albicans is a classic opportunistic pathogen here. It's actually a normal part of the human microbiome. It usually exists perfectly well as harmless, budding yeast cells.
Just hanging out.
Right. But under specific environmental stressors like changes in pH, temperature, or if your immune system gets suppressed, it undergoes a massive phase transition. It stops budding and starts projecting invasive hyphae.
So it essentially turns into a mold so it can physically penetrate through your tissue boundaries.
Exactly. And it gets even more dramatic with the primary deadly pathogens like Histoplasma capsulatum or Blastomyces dermatitis. These are thermally dimorphic.
Meaning temperature is the trigger.
Yes. Out in the environment, sitting in damp soil or like bird guano at room temperature, they exist as molds. They weave their hyphal threads, they release spores, but the moment you inhale those spores and they hit the 37 degrees Celsius furnace of your lungs, boom.
They transform.
Yeah, the temperature shift acts as a direct molecular trigger. It alters the folding of specific regulatory proteins, activating a cascade of gene expression, often involving specific kinase pathways like DRK1.
And so what happens to the fungus?
The fungus immediately aborts its filamentous growth and transforms into a parasitic yeast. And it has to do this because the hyphal mold form would be rapidly destroyed by mammalian macrophages. But the yeast form, it can actually survive inside our immune cells.
Wow. It completely upends how we define these organisms. Like, the boundary between mold and yeast isn't an evolutionary wall, it's just a highly calibrated genetic toggle switch.
Yeah, and understanding that biochemical switch changes the entire paradigm for how infectious disease specialists have to approach diagnostics and treat severe mycosis. I mean, you are dealing with a moving target.
Which is wild. But, it's easy for us to sit here right now analyzing molecular temperature triggers and gene expression cascades. Let's step back to a time when scientists didn't even know these cells were alive.
Oh, the 19th century fermentation debate. It is legendary.
Truly. Proving that yeast was a biological organism, let alone a shapeshifter, sparked one of the most vicious intellectual brawls in scientific history. Because molds were relatively undisputed, right? Since they're macroscopic, you can see them.
Right. In the early 1800s, mycologists like Elias Magnus Fries were already classifying thousands of visible mold species. But yeast remained totally invisible.
Until the 1830s, when microscopy improved enough that researchers like Theodore Schwann and Charles Cagnard de la Tour started looking really closely at the active froth in fermenting beer and wine.
And they observed these tiny asymmetric spheres budding and multiplying. They realized, oh, the froth is actually a living culture. Schwann hypothesized that fermentation wasn't just some passive chemical reaction, but an active biological metabolism performed by these living cells consuming the sugar.
And the dominant chemists of the era absolutely lost their minds over this.
Yeah.
Justus von Liebig and Jöns Jacob Berzelius, who were like the undisputed heavyweights of 19th century chemistry, they aggressively insisted fermentation was purely a process of chemical decomposition.
Yeah, Liebig actually published papers mocking Schwann. Liebig pushed this strictly mechanistic model. He theorized that decaying organic matter created molecular vibrations.
Vibrations.
Yeah, he thought that when dead yeast decayed, it transferred kinetic energy to the sugar molecules, physically shaking them apart into alcohol and carbon dioxide. To them, the yeast was just chemical detritus.
Wait, hold on, though. Why were the chemists so fiercely opposed to this? Bubbling sugar seems like incredibly low stakes for a massive, decade-long scientific feud. Why did it matter so much to Liebig if the yeast was alive or if it was just vibrating chemicals?
If we connect this to the bigger picture, it was not about beer at all. It was a philosophical war over vitalism.
Okay, vitalism, meaning what exactly?
For decades, Chemists had fought to prove that the universe was governed by predictable, observable chemical laws. They were trying to pull science out of the dark ages of mysticism and these elusive, vital forces of life.
Huh.
So to concede that a magical, invisible living cell was required to execute a chemical transformation, it felt like a massive regression to them. They wanted a universe reducible entirely to physics and chemistry.
So they were basically defending their entire scientific paradigm.
Exactly, but they were wrong. The debate raged for 2 whole decades until Louis Pasteur finally settled it between 1857 and 1866.
Right, Pasteur, the heavy hitter.
He executed these beautifully designed, rigorous experiments. Using his famous swan neck flasks, he demonstrated that if you boiled the liquid to kill the yeast or physically filtered the cells out, the fermentation completely ceased, no matter how much chemical precursor you had.
He proved definitively that fermentation was cellular metabolism.
Yes, and the implications of that proof just cascaded through modern history, because Pasteur took that exact logical framework, the idea that specific microscopic living organisms cause specific macroscopic biochemical effects, and he applied it to human pathology.
Oh, wow, that is the direct bridge to the germ theory of disease.
Precisely. The methodological proof that yeast was alive fundamentally birthed modern microbiology and infectious disease medicine.
So Pasteur proved we can't trust what we can't see. But the crazy part is, even when we can see a fungus, when it is growing right in front of us as this massive fuzzy web, our eyes still lie to us today. Which brings us to the phenomenon of mistaken identities.
Yes.
Let's talk about Phytophthora infestans.
A historically devastating organism.
Yeah, this is the pathogen responsible for the Irish potato famine in the 1840s. Anton de Berry, who is widely considered the father of plant pathology, did the foundational work proving this organism caused the late blight, and for well over a century, the entire scientific community studied it as a fungal mold.
Because visually, it behaves exactly like one. It grows filamentous hyphae, it forms a mycelium, it releases spores.
But if you sequence its genome today, Phytophthora is not a mold. It's not even a fungus. It's an oomycete.
Right.
It belongs to a completely different evolutionary lineage, the Stramatoiles, making it more closely related to diatoms and brown algae than to any fungus.
Yeah, it's incredible.
But how does an organism genetically related to kelp end up looking and behaving exactly like a terrestrial fungus?
Well, it is one of the most striking examples of convergent evolution in biology. The filamentous hyphal growth shape is simply an incredibly efficient geometric solution for foraging nutrients in a solid substrate.
Like the potatoes.
Exactly.
Yeah.
So 2 entirely different branches of the tree of life, separated by over 1 billion years of evolution, just independently engineered the exact same blueprint.
It's basically like looking at a shark and a dolphin.
Yeah.
Right? One's a fish, one's a mammal. But they both evolved the exact same streamlined, torpedo-shaped body with dorsal fins, because that's just the most efficient way to move through water.
Yeah.
Fungi and oomycetes both evolved the mycelial shape because it's the most efficient way to eat.
That's spot on, and that visual disguise is flawless. It completely fooled taxonomists. But, this visual bias doesn't just create academic confusion, it drives massive real-world panics, too.
Like what?
Consider the intense media hysteria in the late 1990s and early 2000s surrounding toxic black mold, specifically Stachybotrys chartarum.
Oh man, I remember this vividly. It was a massive cultural phenomenon. There was this terrifying cluster of infant lung pulmonary hemorrhage cases in Cleveland, and early reports strongly suggested household black mold was the primary culprit. I mean, every homeowner was suddenly tearing out their drywall in sheer panic.
Yeah, but the media narrative completely outpaced the actual epidemiology. When the CDC and independent researchers rigorously reevaluated the data, the causal link disintegrated.
Wait, really? The mold didn't cause it?
The epidemiological evidence was highly confounded by other variables like environmental tobacco smoke, other bacterial contaminants, and just water damage in general. The CDC actually had to formally walk back their initial conclusions, stating there was no proven association between Stachybotrys and the infant pulmonary hemorrhages.
Which is a crucial lesson in confounding variables. I mean, the presence of mold indicates water damage, and water damage brings a whole host of other issues. But labeling the mold itself as this lethal, toxic assassin was a massive overreach.
Exactly. Now, that does not mean indoor mold is harmless. It can absolutely trigger severe asthma, allergic rhinitis, and it poses genuine risks to immunocompromised individuals. But the sensationalized archetype of this universal, deadly, toxic black mold lurking behind every wall, that was largely a fiction generated by visual fear rather than cellular mechanics.
However, When the biological and environmental conditions align perfectly, filamentous molds really can become apex predators. Let's look at the devastating surge in mucormycosis, what the press dubbed black fungus in India during the 2021 COVID-19 wave.
Yeah, that was awful.
That was driven by actual mold-formed fungi, primarily Rhizopus species, right?
Yes. And understanding the mechanism behind that outbreak is critical. Because Rhizopus molds are ubiquitous, honestly, you are likely inhaling their spores right now.
Comforting.
Right. But they usually pose zero threat to a functional immune system. The mucormycosis surge occurred because of a perfect storm of host vulnerabilities.
So what happened?
Well, patients were receiving high-dose corticosteroids to combat COVID inflammation. That heavily suppressed their immune responses, and simultaneously it spiked their blood glucose levels.
And a lot of these patients had uncontrolled diabetes to begin with, according to the sources.
Exactly. That combination induces a state called diabetic ketoacidosis, which actually lowers your blood pH. And that acidic environment causes iron to unbind from transport proteins in the blood, essentially flooding the tissue with free iron.
And the mold feeds on that.
Yes. Rhizopus fungi thrive on free iron and high glucose. The mold didn't suddenly mutate into a superpathogen. The human host environment just shifted to perfectly accommodate its rapid hyphal growth strategy. And once established, those filaments invade blood vessels, causing really rapid tissue necrosis.
Man, that is opportunistic biology at its most brutal. So what does this all mean for us? We started out talking about the fuzzy orange in your fridge and the foam on your beer, but the mechanics of these organisms dictate the mechanics of how we live alongside them.
They absolutely do.
Like if you're an infectious disease doctor, understanding if a patient's immune system is failing against the localized yeast or an invasive vascular penetrating mold changes the entire treatment protocol.
Or even if you're a food scientist, you might deliberately cultivate a mold like Aspergillus oryza to ferment soybeans into soy sauce, leveraging its powerful exoenzymes.
Right.
But simultaneously, you have to run rigorous chromatography tests on your grain silos to ensure Aspergillus flavus hasn't taken root, because that visually similar mold synthesizes aflatoxins, which are highly potent hepatic carcinogens.
So you really cannot just rely on the label mold.
Yeah.
It comes down to the precise genetic species, the specific metabolic byproducts, and of course, the state of your own immune system.
Exactly. And if I can leave you with one final thought to mull over, it brings us back to those thermal shapeshifters we discussed earlier.
Oh, the dimorphic fungi.
Right, the ones like histoplasma that use temperature as a genetic toggle switch.
The microscopic werewolves, as I like to call them.
Yes. So right now, human body heat, our 37 degrees Celsius internal temperature acts as a massive thermal barrier. It is simply too hot for the vast majority of environmental fungal species to survive inside us. Our endothermy is a primary evolutionary defense mechanism against fungal infection.
Okay, but the global climate is shifting.
Exactly. As the ambient temperature of the environment rises due to climate change, the soil is getting hotter. The baseline thermal stress on environmental fungi is increasing.
Oh, I see where this is going.
Right. Could these ambient molds slowly adapt and evolve heat tolerance? Because if environmental fungi adapt to thrive at higher baseline temperatures out in the wild, that effectively shrinks the thermal barrier that currently protects our 37-degree bodies.
Wow.
We might be looking at a future where a continually warming world introduces the human immune system to an entirely new catalog of fungal pathogens that were previously just kept at bay by the cold.
That is deeply unsettling, but honestly, it's a perfect illustration of how dynamic biology really is. So the next time you spot that fuzzy patch on a forgotten piece of fruit, do not just see decay. See an ancient, highly evolved survival engine that has confused taxonomists, ignited philosophical wars over the nature of life, and is constantly adapting to the environment around it. Keep questioning what you see, and we will catch you on the next deep dive.